This is Throop Garden, a little stream and waterfall with turtles and goldfish. We're looking east, and there are engineering buildings at the left and at the right. Hello, the next two lectures provide you with a set of two complementary views on the circuit and cellular and molecular basis for nicotine addiction. Again, we are very much concentrating on the question, what changes occur in the brain in response to chronic exposure to nicotine? Just reminding you that nicotine and acetylcholine act on many of the same receptors. But first, acetylcholine is usually hydrolyzed by acetylcholinesterase, which has a turnover rate of about 10 to the fourth per second. On the other hand, in mouse, nicotine is eliminated much more slowly with a half time of 10 minutes, and it's probably 2 or 3 times greater than that in humans. So the difference in these two rates of elimination are about a factor of 10 to the fifth. And now the EC50, the half maximal point of the dose-response relation. Interestingly enough, at muscle receptors, nicotine acts with an EC50 of several hundred micromolars. Acetylcholine acts with an EC50 of about 45 micromolar. The ratio is about 10, and it's justified to square this because the Hill coefficient approaches 2. And so in muscle, the functional ratio between a concentration of nicotine producing a response and a concentration of acetylcholine producing a response is going to be around 100. That's at muscle nicotine receptors. At neuronal nicotinic receptors, though, acetylcholine and nicotine have roughly equal sensitivities. So acetylcholine acts quite efficiently at neuronal brain nicotinic receptors, not at muscle receptors. And this is, of course, why when a person smokes, his muscles don't twitch. If nicotine were as effective at muscle receptors as at brain receptors, there would be these unpleasant side effects of smoking on the muscles, and people would not smoke. A final difference is that nicotine is highly membrane permeant. Acetylcholine is not. The ratio of those differences is unknown. It's probably greater than a factor of 1,000. And this will come in more importantly in our next lecture. Now, I also want to remind you that nicotine and other drugs of abuse have important effects in an acute sense. And so here is a diagram from a wonderful recent review highlighting the midbrain reward system and surrounding systems such as the thalamus, the nucleus accumbens, the pedunculopontine nuclei further back, and nuclei in the basal ganglia. The VTA, the ventral tegmental area, has both GABAergic and dopaminergic neurons in it. Note the color scheme here, and note the presence of all of the receptors that we have been discussing in this course in the context of recreational drugs and abused drugs. A point that we have not discussed much is that many of these receptors are on presynaptic terminals and help to release transmitter. On the other hand, many of them are also on postsynaptic cells, where we have discussed them in detail. I'm not going to go through the entire list of abbreviations here. It's on the next slide, and this slide will be on the course website. Moving on, though, the challenge that I posed at the beginning of our first lecture on nicotine addiction is, what changes in the brain during chronic exposure to nicotine? Not necessarily what the acute effects are, but what builds up over a period of time, 2 days, a week, a month, because chronic exposure to nicotine, maintained repetitive exposure, is what causes nicotine dependence. Well, now, there are two major theories. I'm going to give you the standard theory today, which would be very comfortable to all neuroscientists. I call it the outside-in molecular mechanism for reasons that will become clear in the next lecture. It is signal transduction triggered by a ligand-gated channel. Now, you'll remember that in our lectures on the G protein coupled pathway, we started with activating a receptor, we went through to a G protein, an effector, and we got an intracellular messenger, which was either calcium or a cyclic nucleotide, which, when maintained long enough, activated a kinase, phosphorylated a protein, and got into the nucleus and activated new genes. Now, it's taken as an article of faith by most neuroscientists that long-term changes in the nervous system, and in fact there's a lot of evidence, involve the activation of new genes. And so a wonderful model and example of this is the NMDA receptor, undoubtedly responsible for a great deal of learning and memory, NMDA type glutamate receptor. And NMDA type glutamate receptors are permeable to calcium, which would then serve as a second messenger, bypassing the G protein pathway, activating the pathway, and leading to activated genes. And many neuroscientists would say that this classical, by now classical, description would presumably also apply to nicotine addiction. And in some cases, it probably does. We know that nicotinic receptors are highly permeable to calcium, as well as to sodium. And so it does not take much of a stretch of the imagination to believe that calcium flux through nicotinic receptors in an appropriate place at an appropriate time could go to the same pathway. And various laboratories have been pursuing this hypothesis. Here is an example from the recent literature of one of the transduction pathways of the sort that you'll see in our recommended textbook, and of the sort that I've already shown you. Nicotinic receptors allow calcium. The calcium activates kinases. The kinases go into the nucleus, activate transcription factors such as CRIB. We get new genes activated. For the cognoscenti, it is also possible that calcium activates the MAP kinase pathway and also might interact with the tyrosine kinase receptor pathway. So this is a very active area of research. And in order to find out the key question, which is which genes get activated, and therefore which new proteins get made, we turn to molecular biology. So I'm going to tell you now about so-called transcriptome analysis. And the latest way to do transcriptome analysis is called RNA-seq. We analyze the entire set of RNA produced in a cell by sequencing its RNA. So we begin with the brain from an experimental animal. We cut slices at 30 micron thickness. We put these slices on a special microscope that is equipped for laser capture microdissection. In laser capture microdissection, the cells are sitting on a special membrane, and when a laser beam hits that membrane, it heats up the membrane enough so that the cells are catapulted into a collection vial, and one can then do molecular biology in those individual neurons. Work like this is being done by my group at Caltech and by many others. So the next step is to purify RNA from those cells to make sure that we are dealing only with the messenger RNA by binding poly-DT to make sure that we have only the polyadenylated RNAs. Fragment the RNA, convert it to cDNA using reverse transcriptase, apply special adapters that are suitable for in-depth sequencing, and then put them on one of the latest sequencing machines and analyze millions of reads that are fairly short. They're only about 25 base pairs long. The bioinformatics in an operation like this is really quite intense. And there are various programs which are named after formalware these days to align the readings to the sequence of the genome, to take account of splice sites, to take these rather short reads, 25 base pairs, assemble full-length transcripts from them, figure out how they correspond to groups of genes that are in the database that have been annotated, and group the genes by which pathways have been activated. And it's really the activation of pathways that is perhaps the most exciting possible outcome of understanding the cellular and molecular basis of nicotine addiction. And so I think we can hope that although the relevant papers are just now appearing, researchers may soon have a complete description of the genes activated by chronic exposure to nicotine in each of the relevant neuronal cell types. And with that, I will go on to the next lecture.